The endoplasmic reticulum is a network of interconnected internal membranes, generally, the largest membrane in a eukaryotic cell, an extensive network of closed, flattened membrane-bounded sacs called cisternae. The endoplasmic reticulum has a number of functions in the cell but is particularly important in the synthesis of lipids, membrane proteins, and secreted proteins. Morphology. The endoplasmic reticulum occurs in three forms: 1. Lamellar form or cisternae, which is a closed, fluid-filled sac, vesicle or cavity called cisternae. 2. Vesicular form or vesicle, and 3. Tubular form or tubules. Cisternae. The cisternae are long, flattened, sac-like, unbranched tubules having a diameter of 40 to 50 micrometers. They remain arranged parallelly in bundles or stakes. RER mostly exists as cisternae, which occur in those cells which have synthetic roles as the cells of the pancreas, notochord, and brain. Vesicles. The vesicles are oval, membrane-bound vacuolar structures having a diameter of 25 to 500 micrometers. They often remain isolated in the cytoplasm and occur in most cells but are especially abundant in the SIR. Tubules. The tubules are branched structures forming the reticular system along with the cisternae and vesicles. They usually have a diameter from 50 to 190 micrometers and occur almost in all cells. The tubular form of ER is often found in SIR and is dynamic in nature, i.e., it is associated with membrane movements, fission, and fusion between membranes of cytocavity network. Types of endoplasmic reticulum. A granular or smooth endoplasmic reticulum. ER with no studded ribosomes makes it smooth in appearance. The adipose tissues, brown fat cells, adrenocortical cells, interstitial cells of the testes and cells of the corpus luteum of ovaries, sebaceous cells, and retinal pigment cells contain only smooth endoplasmic reticulum, SIR. The synthesis of fatty acids and phospholipids takes place in the smooth ER. It is abundant in hepatocytes. Enzymes in the smooth ER of the liver modify or detoxify hydrophobic chemicals such as pesticide and carcinogens by chemically converting them into more water-soluble, conjugated products that can be excreted from the body. High doses of such compounds result in a large proliferation of the smooth ER in liver cells. Granular or rough endoplasmic reticulum. Ribosomes bound to the endoplasmic reticulum make it appear rough. The rough ER synthesizes certain membrane and organelle proteins and virtually all proteins to be secreted from the cell. A ribosome that fabricates such a protein is bound to the rough ER by the nascent polypeptide chain of the protein. As the growing polypeptide emerges from the ribosome, it passes through the rough ER membrane, with the help of specific proteins in the membrane. Newly made membrane proteins remain associated with the rough ER membrane, and proteins to be secreted accumulate in the lumen of the organelle. All eukaryotic cells contain a discernible amount of rough ER because it is needed for the synthesis of plasma membrane proteins and proteins of the extracellular matrix. Rough ER is particularly abundant in specialized cells that produce an abundance of specific proteins to be secreted. The cells of those organs which are actively engaged in the synthesis of proteins such as acinar cells of the pancreas, plasma cells, goblet cells, and cells of some endocrine glands are found to contain rough endoplasmic reticulum, RER, which is highly developed. Common functions of SIR and RER. The endoplasmic reticulum provides an ultrastructural skeletal framework to the cell and gives mechanical support to the colloidal cytoplasmic matrix. The exchange of molecules by the process of osmosis, diffusion, and active transport occurs through the membranes of the endoplasmic reticulum. The ER membrane has permeases and carriers. The endoplasmic membranes contain many enzymes which perform various synthetic and metabolic activities and provide an increased surface for various enzymatic reactions. The endoplasmic reticulum acts as an intracellular circulatory or transporting system. Various secretory products of granular endoplasmic reticulum are transported to various organelles as follows: granular ER-, agranular ER-, Golgi membrane-, lysosomes, transport vesicles or secretory granules. Membrane flow may also be an important mechanism for carrying particles, molecules, and ions into and out of the cells. Export of RNA and nucleoproteins from the nucleus to the cytoplasm may also occur by this type of flow. The ER membranes are found to conduct intracellular impulses. For example, the sarcoplasmic reticulum transmits impulses from the surface membrane into the deep region of the muscle fibers. The sarcoplasmic reticulum plays a role in releasing calcium when the muscle is stimulated and actively transporting calcium back into the sarcoplasmic reticulum when the stimulation stops and the muscle must be relaxed.